This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to this exploration of one of life's most remarkable transformations. Around 470 million years ago, plants took their first brave steps from water onto land, a monumental shift that would forever change our planet. Today, we'll journey through the incredible adaptations that made this possible, from pioneering root systems and water-conducting tissues to protective waxy coatings and revolutionary reproductive strategies. These innovations didn't just allow plants to survive on land, they created the green world we depend on today. Let's discover how plants conquered the terrestrial frontier. Chapter 1. From Water to Land. The transition of plants from water to land stands as one of the most profound events in the history of life on Earth. This evolutionary leap fundamentally transformed our planet, reshaping its atmosphere, climate, and the very surface upon which we walk. Before plants colonized the land, the terrestrial world was a stark and barren landscape, dominated by rock and mineral soil, with only microbial life forming thin crusts in favorable locations. The greening of the continents would change everything, creating soil where none existed, altering atmospheric oxygen levels, moderating climate through moisture cycling, and establishing the foundation for all terrestrial ecosystems. Current scientific understanding holds that all plants, both green algae and land dwellers, are monophyletic, meaning they descend from a single common ancestor. This places all members of the plant kingdom within one great family tree, sharing fundamental characteristics that unite them despite their tremendous diversity. At some pivotal point in evolutionary history, estimated at approximately 480 to 470 million years ago during the Paleozoic era, there was a momentous transition from an aquatic environment to a terrestrial one. This shift from water to land imposed severe constraints on the organisms attempting it, yet it also opened up vast new opportunities. To understand this transition, we must first examine the aquatic ancestors of land plants. Recent phylogenetic analyses, which trace evolutionary relationships through genetic and morphological evidence, have identified these ancestors among a group of green algae called charophytes. These studies have placed the ancestor of land plants close to a common ancestor shared between two orders of charophytes called zygnematotales and coleochaetales. This finding is significant because both of these orders contain species that already live in terrestrial habitats or display the capability to do so. Charophytes represent a diverse group of green algae that includes several different classes, ranging from relatively simple organisms to more complex forms. What makes them particularly interesting is that terrestrialization, the adaptation to terrestrial life, is not a unique trait found only in the charophytes most closely related to land plants. Even less advanced classes, such as Clubsormidiophyceae and Chlorochibophyceae, contain terrestrial species. This widespread occurrence of terrestrial adaptations across multiple charophyte groups suggests something fascinating about the origins of land plants. An alternative hypothesis challenges the traditional view that land plants evolved from aquatic ancestors and then adapted to land. Instead, this perspective proposes that the charophyte ancestors were already living on land and had been doing so for some considerable time before the emergence of what we recognize as true land plants. According to this view, these ancestral organisms had already solved many of the fundamental challenges of terrestrial existence. Subsequent evolution of more complex features, such as the alternating life cycles characteristic of land plants, merely made these organisms more successful in their terrestrial environment, but had nothing to do with the initial terrestrialization itself. Evidence supporting the idea that charophyte ancestors were already terrestrial comes from multiple sources. Morphological and physiological observations of living xerophytes reveal that many species are perfectly capable of surviving in terrestrial or semi-terrestrial habitats. Some species of coleochaete, for instance, are described as aeroterrestrial, living in environments that are neither fully aquatic nor fully terrestrial, but somewhere in between. These organisms can be found growing on damp rocks, in thin films of water, or in areas that alternate between wet and dry conditions. Such habitats would have been abundant along the edges of ancient bodies of water, providing ideal transitional environments for organisms evolving toward a terrestrial lifestyle. The genome of Klebsormidium flaccidum, sequenced in recent years, has revealed primary factors for plant terrestrial adaptation already present in this relatively basal xerophyte. This organism possesses genetic machinery for dealing with terrestrial challenges, suggesting that the toolkit for life on land evolved early in the xerophyte lineage, well before the emergence of true land plants. Novel analyses of both advanced and basal xerophyte biochemistry, transcriptomes, and genomes have revealed a range of terrestrial adaptations that are orthologous to what we observe in land plants, meaning they share a common evolutionary origin. Broad phylogenomic sampling, which examines many genes across numerous species, has helped clarify the relationships among green algae and land plants. The green lineage, as it is often called, includes both the chlorophytes, which are primarily aquatic green algae less closely related to land plants, and the streptophytes, which include the xerophytes and all land plants. Within the streptophytes, the exact branching order of xerophyte groups and their relationship to land plants has been a subject of intensive investigation. Early phylogenetic work identified several possible candidates for the closest living relatives of land plants. The charals, an order of complex green algae with specialized structures and relatively large size, were once considered prime candidates. However, more recent analyses using molecular data have pointed instead toward the zygnometals, the group that includes organisms like Spirogyra, known for their spiral chloroplasts, and various species of pond scum. Some studies have suggested that conjugating green algae, which reproduce through a distinctive process where two cells fuse their contents, might hold the key to understanding land plant origins. Identifying the closest xerophyte relatives of land plants goes beyond mere taxonomy. These organisms serve as living models for understanding what the ancestor of land plants might have been like and what capabilities it might have possessed. Studies of coleochaete have revealed lignin-like compounds and sporopollenin, complex molecules that in land plants serve critical functions in structural support and spore protection respectively. The presence of these compounds in xerophytes suggests that some of the biochemical innovations we associate with terrestrial plants actually evolved earlier in their aquatic or semi-aquatic ancestors. The structure and biochemistry of xerophyte cell walls have provided particularly valuable insights into plant evolution. Research on species like Penium margaritaceum has revealed cell wall components, particularly pectins, that resemble those found in land plants. High-throughput mapping of cell wall polymers has shown that xerophytes possess various polysaccharides previously thought to be unique to land plants, including the primary cell wall polysaccharide rhamnogalacturonin found in bryophytes, lycophytes, and pteridophytes. These findings indicate that the basic architecture of the plant cell wall, which would prove essential for supporting terrestrial plant bodies, was already being developed in aquatic ancestors. The discovery of lignin-like compounds in xerophytes has proven particularly intriguing. This molecule provides rigidity to plant cell walls and is essential for the support of upright stems. Long thought to be an innovation of land plants, research has detected lignin-related epitopes in the cell walls of some xerophytes, suggesting that precursors to this important molecule existed before plants moved onto land. The fossil record provides crucial evidence for dating the colonization of land by plants. The earliest definitive fossils of land plants appear in rocks from the mid-Ordovician period, roughly 470 million years ago. These fossils consist of spores with distinctive features characteristic of land plants. Somewhat later, from the Silurian period, around 420 million years ago, we find fossils of Cooksonia, often cited as the first vascular land plant. Cooksonia was a small, simple plant, lacking true roots or leaves, that grew perhaps a few inches off the ground. Its structure consisted of branching stems tipped with spore-producing structures called sporangia. The fossil record also contains intriguing evidence of even earlier terrestrialization. Thalloid carbonaceous incrustations from the early Paleozoic suggest that the evolution of various embryophyte characteristics occurred asynchronously. Different adaptations appeared at different times rather than all at once in a single evolutionary leap. Some organisms might have possessed certain land plant features while lacking others, creating a mosaic of characteristics during the transition period. This gradual assembly of terrestrial capabilities helps explain why the exact timing and sequence of plant terrestrialization remains somewhat uncertain. The timing of plant colonization of land had profound implications for other life forms. Land plants evolved before land animals, which means that for a considerable period, until dry land was also colonized by animals, no predators threatened plant life. This situation changed as animals emerged from the water and began to feed on the abundant sources of nutrients provided by the established flora. The appearance of herbivorous animals created new selective pressures, driving the evolution of plant defenses, ranging from physical structures like spines and thorns to an impressive array of toxic chemicals. The shift to terrestrial life presented formidable challenges that would drive remarkable evolutionary innovations. Plants had to prevent desiccation, withstand unfiltered ultraviolet radiation, support themselves against gravity without water's buoyancy, and reproduce without water for gamete dispersal. Yet the advantages of land, abundant sunlight, readily available carbon dioxide, and initially the absence of herbivorous predators made the evolutionary investment worthwhile. The colonization of land by plants was not a single event, but rather a prolonged process involving multiple lineages and occurring in stages. Not all groups of plants moved equally far from their aquatic origins. Some species never moved very far from abundant sources of water, whereas others went on to conquer even the driest environments on Earth. This variation reflects different evolutionary strategies and the diverse solutions that evolution found to the challenges of terrestrial life. Early land plant communities likely existed in marginal habitats along the edges of bodies of water, in areas that were periodically flooded, or in locations where moisture remained abundant even if standing water was not always present. These transitional zones between aquatic and terrestrial environments would have provided ideal conditions for organisms adapting to life on land. The role of fungi in plant colonization of land deserves particular attention. Evidence suggests that the association between plant roots and mycorrhizal fungi, a mutualistic relationship where fungi help plants acquire nutrients while receiving carbohydrates in return, evolved very early in land plant history. This partnership may have been essential for the success of early land plants, helping them acquire nutrients from rock and primitive soils. The colonization of land by plants and fungi appears to have been intertwined, with each group facilitating the success of the other. Sunlight represented the first major advantage that drew organisms to colonize land. While water supports aquatic life, it also acts as a filter, altering the spectral quality of light that penetrates to various depths. Photosynthetic organisms in water must cope with diminishing light intensity as depth increases, and the wavelengths of light available change with depth as water selectively absorbs different parts of the spectrum. On land, by contrast, sunlight was abundant and unfiltered by water, offering tremendous potential for photosynthesis to organisms that could survive the terrestrial environment. On land, by contrast, sunlight was abundant and unfiltered by water, offering tremendous potential for photosynthesis to organisms that could survive the terrestrial environment. Carbon dioxide availability represented another significant advantage. This gas, essential for photosynthesis, diffuses much faster in air than in water. Aquatic photosynthetic organisms must rely on the relatively slow diffusion of dissolved carbon dioxide through water, or they must have mechanisms to concentrate it. Terrestrial plants, once they evolved appropriate structures, could access atmospheric carbon dioxide much more readily, potentially allowing for higher rates of photosynthesis given adequate water and nutrients. The greening of the land by plants had cascading effects on the entire planet. Plants fundamentally altered the atmosphere through their photosynthetic activity, contributing to the buildup of oxygen that had begun with aquatic photosynthetic organisms but now accelerated as terrestrial plant communities expanded. This oxygen enrichment had profound implications for the evolution of animal life, enabling the development of organisms with high metabolic rates that required abundant oxygen. Beyond atmospheric changes, terrestrial plants transformed the physical environment of the land surface. Plant roots, working in concert with fungi and bacteria, began to break down rock into soil, creating an entirely new type of habitat. This soil formation process, essentially non-existent before plants colonized land, would become one of the most important biogeochemical processes on the planet. Soil provides habitat for countless organisms and serves as the medium for nutrient cycling that sustains terrestrial ecosystems. Plants also changed patterns of water movement and retention on land. By taking up water through roots and releasing it through leaves, plants became major players in the water cycle. Their presence increased the amount of water vapor returned to the atmosphere through transpiration, affecting local and regional climate patterns. Plant cover also reduced erosion, helping to retain soil and nutrients that might otherwise be washed away. The evolutionary innovations that enabled plant terrestrialization did not all appear simultaneously. Some adaptations evolved early and were present in charophyte ancestors, while others emerged specifically in response to terrestrial challenges. The transition involved both the refinement of existing capabilities and the evolution of entirely new structures and processes. Understanding this evolutionary sequence requires examining both the characteristics shared between charophytes and land plants and the novel features that distinguish land plants from their algal relatives. Comparative analyses of charophyte biochemistry have revealed surprising similarities to land plants. The presence of cell wall components like pectins and hemicelluloses in charophytes demonstrates that fundamental aspects of plant cell wall architecture predate the colonization of land. Similarly, the occurrence of complex polysaccharides like rhamnogalacturonin in various plant groups suggests ancient origins in plant evolution. The evolution of land plants represents a transition not just in where organisms lived, but in how they functioned. The glycosyl transferase repertoire, the set of enzymes responsible for building complex carbohydrates, expanded and diversified as plants adapted to terrestrial life. Comparative studies of these enzyme families across the plant kingdom reveal patterns of gene duplication, loss, and functional divergence that track the evolution of new capabilities. Evidence for land plant cell wall biosynthetic mechanisms appearing in charophyte green algae suggests that the genetic toolkit for building terrestrial plant bodies was assembled gradually over evolutionary time. As we examine the evolutionary path from aquatic algae to terrestrial plants, we must remember that evolution does not proceed with foresight or purpose. Charophytes that possessed characteristics pre-adapting them for terrestrial life did not develop those characteristics in order to colonize land. Rather, these features likely served functions in their original aquatic or marginal habitats and only later proved advantageous when organisms encountered terrestrial conditions. This principle, called exaptation, reminds us that evolution works by modifying existing structures and capabilities, not by designing organisms from scratch for new environments. The colonization of land by plants opened up an entirely new realm for biological diversification. Opportunities for specialization in different terrestrial habitats, from the wettest to the driest, from the hottest to the coldest, drove the evolution of tremendous diversity we see in land plants today. From tiny mosses growing in rock cracks to giant sequoias towering hundreds of feet in the air, From plants that complete their entire life cycle in weeks to those that live for thousands of years, the descendants of those first colonizers have explored nearly every possibility that terrestrial life offers. Understanding how plants conquered land provides crucial context for appreciating the diversity and adaptations of modern plant life. It reminds us that the familiar features of land plants, from roots that anchor them in soil to leaves that capture sunlight, from woody stems that support towering trees to seeds that can survive years of dormancy, all represent solutions to challenges that first arose when organisms began the transition from water to land hundreds of millions of years ago. These evolutionary innovations transformed our planet and made possible the rich terrestrial ecosystems on which all land animals, including humans, ultimately depend. Chapter 2: The Gauntlet of Terrestrial Life. The transition from aquatic to terrestrial environments presented organisms with a formidable array of challenges, each potentially fatal if not adequately addressed. To appreciate the magnitude of these obstacles, we must first understand what the aquatic environment provides that the terrestrial environment does not. Water is not merely the medium in which aquatic organisms live, it is fundamental to nearly every aspect of their biology. The shift to life on land required solving multiple, interconnected problems simultaneously, a feat that took millions of years of evolutionary experimentation and refinement. The most immediate and perhaps most severe challenge facing early terrestrial plants was the constant threat of desiccation. The interior of a cell has been aptly described as a watery soup, and this description captures an essential truth about life itself. In this aqueous medium, small molecules dissolve and diffuse freely, and the vast majority of chemical reactions that sustain metabolism take place. Every enzyme, every biochemical pathway, every fundamental process of life depends on the presence of water. The chemical reactions of photosynthesis, the process by which plants convert light energy into chemical energy, require water not just as a raw material, but as the medium in which the reactions occur. Even the physical structure of proteins, the molecular machines that carry out cellular functions, depends on their interaction with surrounding water molecules. For an organism living in a pond, lake, or ocean, maintaining hydration presents no particular difficulty. Water is everywhere, surrounding every surface, available in effectively unlimited quantities. The body of an aquatic organism can freely exchange water with its environment, and internal water content remains relatively constant, without any special mechanisms or structures. But the moment an organism emerges into the air, this situation changes dramatically. Air, even humid air, represents a fundamentally desiccating environment compared to water. The difference in water vapor pressure between the inside of plant tissues, which must remain hydrated, and the surrounding air creates a driving force for water loss that operates continuously. The danger of drying out is not merely that a plant might become uncomfortably dry in the way that we might feel thirsty. For most organisms, severe desiccation means death. When cells lose too much water, their proteins denature and lose function. Their membranes rupture, and their biochemical machinery grinds to a halt. The threshold between survivable water loss and lethal desiccation can be surprisingly narrow. This meant that any organism attempting to colonize land had to solve the water problem or perish in the attempt. The challenge was compounded by the fact that different parts of a plant face different degrees of desiccation stress. Even when parts of a plant are close to a source of water, perhaps with roots in moist soil or submerged in a stream, the aerial structures remain prone to drying out. Leaves, in particular, face an acute dilemma. They must be positioned to capture sunlight for photosynthesis, which often means being exposed to air and wind, the very conditions that accelerate water loss. The larger the surface area a plant exposes to sunlight, the more photosynthesis it can carry out, but also the more surface area it exposes to the desiccating effects of air. Water loss from plants occurs primarily through evaporation, a purely physical process driven by the difference in water vapor concentration between the plant's interior and the surrounding air. On a hot, dry, windy day, the rate of water loss can be staggering. A large tree might transpire hundreds of liters of water in a single day. For early land plants, which lacked many of the sophisticated water management systems of modern plants, the rate of water loss would have been similarly severe relative to their size. This created an enormous constraint on where these plants could survive. They had to remain close to reliable water sources and could not venture into areas where water was scarce or seasonal. Yet plants could not simply seal themselves off from the environment to prevent water loss, because they faced another requirement that worked directly against water conservation. Photosynthesis requires carbon dioxide, which must be obtained from the atmosphere. Acquiring carbon dioxide means providing some pathway for gas exchange between the plant's interior and the air. Any opening that allows carbon dioxide to enter also provides a route for water vapor to escape. This created a fundamental trade-off that plants still grapple with today: the need to balance carbon dioxide uptake for photosynthesis against water loss through the same openings. The temperature extremes of terrestrial environments added another dimension to the desiccation challenge. Water moderates temperature fluctuations because of its high heat capacity. It takes substantial energy to heat or cool water. Aquatic organisms experience relatively stable temperatures compared to their terrestrial counterparts. But on land, daily and seasonal temperature swings can be dramatic. The desert exemplifies this challenge in its most extreme form, with soaring heat during the day that can exceed 40 degrees Celsius and sub-zero temperatures at night. These temperature fluctuations affect water loss rates, with higher temperatures generally accelerating evaporation. They also affect the physiology of organisms more directly, as enzymes and other cellular components function optimally only within certain temperature ranges. Early land plants dealt with the water challenge through different strategies that reflected different evolutionary solutions. Some plants developed what we might call tolerance, an ability to withstand severe desiccation and recover when water became available again. Mosses exemplify this strategy beautifully. These small plants can dry out to a brown and brittle mat that appears completely dead. Their cells lose most of their water content, their metabolism shuts down, and they enter a state of suspended animation. But this is not death. As soon as rain falls or a flood makes water available, mosses absorb moisture with remarkable speed and are restored to their healthy green appearance within hours. This tolerance strategy allowed mosses to colonize environments where water availability was intermittent, expanding the range of habitats where plant life could exist. Other early plants adopted a different approach, avoidance rather than tolerance. These plants colonized environments with high humidity, where droughts were uncommon. Ferns represent this strategy. Considered an early lineage of plants in terms of their evolutionary position, ferns thrive in damp and cool places, such as the forest understory. By selecting habitats where water is reliably abundant, ferns could avoid the severe desiccation stress that would prove lethal to them. This strategy limited where they could live, but allowed them to maintain relatively simple water management systems. Still later in plant evolution, a third strategy emerged: resistance. Rather than tolerating desiccation or avoiding dry environments, some plants evolved mechanisms to minimize water loss to such an extent that they could survive in extremely dry environments. Cacti and other desert plants exemplify this resistance strategy, possessing elaborate adaptations that drastically reduce evaporative water loss while still allowing photosynthesis to continue. The second major challenge of terrestrial life involved radiation. Water absorbs and filters ultraviolet radiation, particularly the highly energetic UVB wavelengths that are especially harmful to living organisms. In an aquatic environment, even relatively shallow water provides significant protection against UV radiation. The deeper an organism lives, the more complete this protection becomes. But air provides no such filtering. The moment an organism emerges from water into air, it becomes exposed to the full intensity of solar UV radiation. This exposure creates serious problems because UV radiation damages biological molecules, particularly DNA. When UV photons strike DNA, they can cause adjacent thymine bases to link together abnormally, forming thymine dimers that distort the DNA structure and interfere with its replication and transcription. If not repaired, these lesions can lead to mutations or cell death. For single-celled organisms, this might mean the death of the individual. For multicellular organisms, UV damage can kill cells, disrupt tissues, and over time lead to the accumulation of harmful mutations. The danger is particularly acute for photosynthetic organisms because, by their very nature, they must position themselves to capture light. The same structures that absorb the visible wavelengths needed for photosynthesis are also exposed to the UV radiation that accompanies sunlight. Plants cannot simply hide from UV radiation the way that some animals might seek shelter. They must function in full sun to carry out photosynthesis efficiently. This created an inescapable dilemma requiring evolutionary solutions that could provide protection without blocking the light needed for photosynthesis. Beyond the direct damage to DNA, UV radiation can also generate reactive oxygen species, highly reactive molecules that damage cellular components, including lipids, proteins, and nucleic acids. These reactive oxygen species act as a secondary effect of UV exposure, extending the harm beyond just direct photon damage. The combination of direct DNA damage and oxidative stress from reactive oxygen species made UV radiation one of the most significant hazards of terrestrial life. Early plants that colonized land had to evolve protective mechanisms against UV damage. The evolution of biosynthetic pathways for the synthesis of protective compounds became essential. Flavonoids and other related molecules, which are pigments that absorb UV wavelengths of light, evolved to protect the aerial parts of plants from photodynamic damage. These compounds act as a sort of molecular sunscreen, absorbing harmful UV photons before they can damage sensitive cellular components. The presence of these protective molecules represents one of the key innovations that made terrestrial plant life possible. The challenge of UV protection also affected plant reproductive structures. Spores, the reproductive cells of early land plants, required special protection because they often traveled through the air and were exposed to intense UV radiation during dispersal. The evolution of sporopollenin provided this protection, becoming a crucial component of spore walls. Its ability to shield against UV radiation made it ideally suited for protecting reproductive cells. The third major challenge of terrestrial life involved structural support. In water, buoyancy provides a supportive force that helps organisms maintain their shape and position. The density of living tissues is similar to the density of water, which means that aquatic organisms are nearly weightless in their environment. This allows aquatic plants to grow upward toward light sources without requiring substantial structural support. The flexible stems and leaves of kelp and other seaweeds, for instance, can extend many meters in length while remaining delicate and pliable because water supports their weight. On land, this support of buoyancy disappears. Gravity becomes a formidable enemy, pulling constantly downward on every part of an organism. For a plant emerging from water, this meant that structures that could float freely in an aquatic environment would simply collapse under their own weight in air. Early land plants had to develop mechanisms for mechanical support that could counteract the force of gravity, or they would be limited to growing as flat mats pressed against the ground. This structural challenge intersected with the imperative to compete for light. Early land plants that remained low to the ground faced intense competition for sunlight. Plants growing as low mats, only a few inches off the ground, would shade each other, with those on top capturing the majority of available light. This created strong selective pressure for height. By developing shoots and growing taller, individual plants could capture more light, giving them an advantage over their competitors. But growing taller required ever more sophisticated structural support. The evolution from prostrate mats of vegetation to upright stems represents one of the major transitions in plant evolution. Because air offers substantially less support than water, plants had to incorporate rigid structural molecules into their stems, particularly lignin. When deposited in cell walls alongside cellulose, lignin creates a composite material combining tensile strength with compression resistance, similar to the rebar in reinforced concrete. This rigidity had to be balanced against flexibility. Completely rigid structures would be brittle and likely to break under stress from wind or other forces. Plants needed materials that could provide strength and support while still allowing some degree of bending and movement. The structural challenge became more acute as plants grew larger. A plant a few centimeters tall requires far less structural support than one that reaches meters into the air. As plant size increased through evolutionary time, from the small early land plants to the towering trees that would eventually dominate many terrestrial landscapes, the solutions to the structural support problem had to become increasingly sophisticated. The development of wood and the evolution of tree-like growth forms represent the culmination of this evolutionary trajectory. But they build on structural innovations that first appeared in much smaller, earlier plants. Growing taller also created a transport problem that small plants could avoid. Simple diffusion, the passive movement of molecules from areas of high concentration to areas of low concentration, works adequately over short distances. A plant only a few cells thick can rely on diffusion to move water, minerals, and nutrients throughout its body. But diffusion is far too slow to move materials through a plant that stands meters tall. The evolution of vascular tissue, specialized conducting tubes that could transport water upward from roots and distribute sugars produced by photosynthesis throughout the plant, became essential for height. These transport systems, working in concert with lignified structural support, allowed plants to reach sizes that would have been impossible through diffusion alone. The structural demands of terrestrial life also affected plant root systems. In water, roots primarily served to anchor the organism. But on land, roots took on additional critical functions. They had to absorb water from soil and transport it to aerial parts of the plant. They had to acquire mineral nutrients, often scarce in primitive soils. The evolution of true roots, as opposed to the simple rhizoids of early land plants, represented another major innovation, driven by the structural and physiological demands of terrestrial life. The challenge of nutrient acquisition on land proved particularly acute. Early soils lacked the organic matter that enriches modern soils, and many essential minerals existed in forms that plant roots could not easily access. The association between plant roots and mycorrhizal fungi, a mutualistic partnership where fungi help plants acquire nutrients while receiving carbohydrates in return, may have been crucial in the initial colonization of land. This partnership may have been essential for early plants attempting to establish themselves in nutrient-poor terrestrial environments. The continuous growth capacity of plants represented another major adaptation for terrestrial life. Coupled with the structural support provided by lignin and the transport capabilities of vascular tissue, enabling plants to achieve sizes and complexity impossible in their aquatic ancestors. The fourth major challenge involved reproduction. In aquatic environments, many organisms reproduce by releasing gametes into the water, where they can swim to find each other. The flagellated sperm of algae and many other aquatic organisms are well adapted to this mode of reproduction, using their whip-like flagella to propel themselves through water toward eggs. This reproductive strategy works well in water but fails completely in air. Sperm cannot swim through air, and the distances involved on land often far exceed what swimming gametes could traverse even in water. Early land plants faced the problem of how to get male gametes to female gametes using new strategies, because swimming was no longer a practical option except over very short distances in films of water. This constraint meant that some early land plants remained dependent on water for reproduction even after colonizing land for their vegetative growth. They could only successfully reproduce when water was present, limiting them to moist habitats or restricting their reproduction to wet seasons. The protection of gametes and zygotes presented another reproductive challenge. In water, gametes are surrounded by a medium that prevents them from drying out and provides some protection against environmental stresses. But on land, any reproductive cell exposed to air faces the same desiccation risks as vegetative tissues. Both gametes and zygotes required protection from drying out if reproduction was to succeed. This led to the evolution of structures that could house and protect reproductive cells, keeping them in moist environments even when surrounded by air. The developing embryo represented a particular challenge. After fertilization, the young sporophyte, as the diploid generation is called, must develop from a single-celled zygote into a multicellular organism. During this vulnerable developmental period, the embryo requires protection from desiccation and other environmental hazards. In aquatic plants, the embryo develops in water, but terrestrial plants needed new mechanisms to shelter the embryo. The evolution of structures that could provide protection and nutrients to the developing embryo became a defining characteristic of land plants, so much so that they are sometimes called embryophytes to emphasize this feature. Dispersal of reproductive cells posed yet another problem. Many aquatic organisms rely on water currents to distribute their offspring, but terrestrial plants needed alternative dispersal mechanisms. Some early land plants released spores into the air, relying on wind for dispersal. But this required that the spores be light enough to be carried by air currents and tough enough to survive the journey. Other reproductive strategies involved the evolution of seeds, packages that not only protected the embryo, but also provided it with stored nutrients to support its initial growth after germination. All of these challenges, desiccation, UV radiation, structural support, and reproduction without water, were interconnected. Solutions to one problem often created new constraints or opportunities related to the others. For instance, structures that provided UV protection might also help reduce water loss, or mechanisms for structural support might be adapted to protect reproductive tissues. The evolutionary solutions that emerged represent integrated responses to the multiple challenges of terrestrial life, rather than isolated adaptations to individual problems. The severity of these challenges meant that the colonization of land by plants was not a rapid conquest, but a gradual process. Early terrestrial plants remained close to water and reproduced only under moist conditions. They were small, structurally simple organisms that could tolerate or avoid the worst stresses of terrestrial life, but could not yet thrive in more challenging environments. The evolution of more sophisticated adaptations proceeded over tens of millions of years, with each innovation opening new possibilities for where and how plants could live. Some lineages remained conservative, continuing to rely on strategies that worked in moist environments. Others became increasingly adventurous, developing adaptations that allowed them to colonize progressively drier and more challenging habitats. Understanding these challenges provides essential context for appreciating the adaptations that plants evolved. Every structural feature, every biochemical pathway, every aspect of plant life history can be understood as a response to one or more of these fundamental challenges. The waxy surfaces of leaves, the pores that control gas exchange, the rigid stems that hold plants upright, the elaborate life cycles that protect embryos, all of these and countless other plant features represent solutions to the challenges we have explored. In the chapters that follow, we will examine the specific structural and reproductive adaptations that allowed plants to overcome these obstacles and eventually dominate terrestrial ecosystems across the globe. Chapter 3: The Terrestrial Toolkit. The structural innovations that enabled plant life on land represent one of evolution's most remarkable engineering achievements. Yet these innovations did not appear suddenly or simultaneously. Recent research has revealed a surprising fact. Many of the molecular and structural features we associate with terrestrial plants actually evolved earlier in xerophyte algae that were the aquatic or semi-aquatic ancestors of land plants. The transition to land involved both the refinement of pre-existing capabilities and the evolution of entirely new structures, assembled over tens of millions of years into an integrated system. Understanding when and how these adaptations appeared requires abandoning the traditional view of plant terrestrialization as a dramatic leap from water to land. The xerophyte ancestors of land plants were not purely aquatic organisms that suddenly faced terrestrial challenges. Many lived in marginal habitats, in thin films of water on rocks, in temporary pools, or in areas that alternated between wet and dry conditions. These environments already presented some terrestrial challenges, and natural selection had already begun shaping solutions. Genetic analyses of modern xerophytes have revealed genes for UV protective compounds, cell wall components similar to those in land plants, and even lignin-like molecules. The toolkit for terrestrial life was being assembled piece by piece in organisms that remained primarily aquatic. The waxy cuticle likely represents the earliest of the major structural innovations, appearing even in some xerophyte ancestors where it may have served functions other than preventing water loss. This thin layer, composed primarily of cutin, a complex polymer built from fatty acids and other hydrophobic molecules, along with embedded waxes made of very long-chain fatty acids, creates a barrier that water molecules cannot easily penetrate. This thin layer, composed primarily of cutin, forms a three-dimensional polymer network into which wax components are embedded. This dual-component system provides robust protection against desiccation while maintaining the mechanical properties needed to cover plant surfaces. In the earliest land plants, the cuticle remained relatively thin. The bryophytes, retaining many ancestral characteristics, possess cuticles that provide modest protection adequate for the moist environments these plants inhabit. A thin cuticle allows some gas exchange directly through the surface while still reducing water loss compared to having no cuticle at all. As plants colonized progressively drier environments, selection favored increasingly elaborate cuticles. Desert plants possess cuticles of remarkable thickness and complexity, with multiple wax layers creating barriers so effective that water loss is reduced to a tiny fraction of what would occur through unprotected surfaces. The cuticle's dual function merits emphasis. Beyond waterproofing, it provides partial protection against ultraviolet radiation. The waxy molecules absorb certain UV wavelengths, acting as a physical shield between delicate cellular machinery and the harsh radiation environment of the terrestrial surface. This protection proved critical because early land plants faced intense UV exposure as soon as they emerged from water's protective filtering. A single innovation addressing two major terrestrial challenges simultaneously helps explain why the cuticle appears universally in land plants. The evolution of stomata followed as a necessary response to the cuticle's success. A barrier that prevents water from escaping also prevents gases from entering or leaving the plant. Carbon dioxide, the raw material for photosynthesis, cannot diffuse through the waxy cuticle, nor can oxygen escape. The solution came through microscopic pores that penetrate the cuticle and epidermis, creating controlled openings between the plant's interior and the atmosphere. Each stoma consists of a pore surrounded by two specialized guard cells that change shape to open or close the pore through an elegant mechanism involving osmotic pressure. When guard cells actively pump potassium ions into their interior, water follows by osmosis, causing the cells to swell. The construction of guard cells, with cell walls thicker on one side than the other, means that swelling causes them to bow outward, creating an opening between them. When guard cells reverse the process, pumping potassium out and losing water, they become flaccid and press together, closing the pore. This ability to regulate gas exchange represented a crucial innovation, allowing plants to balance the competing demands of carbon dioxide acquisition and water conservation. Stomata appear in nearly all land plants, with liverworts as the notable exception. The distribution and density of stomata vary remarkably across species, reflecting different adaptations to different environments. Desert plants typically have fewer stomata per unit area compared to plants from wetter environments, and some place their stomata in sunken pits, creating more humid microenvironments that further reduce evaporation. The evolution of vascular tissue and true roots occurred together, representing the third major innovation. While small plants can rely on diffusion to move water and nutrients through their tissues, diffusion works only over short distances. A plant more than a few centimeters tall requires a more efficient transport system. The solution came through tube-like cells specialized for long-distance transport. Xylem tissue transports water and dissolved minerals upward from roots to leaves, while phloem tissue transports sugars produced by photosynthesis throughout the plant. The structure of xylem reveals elegant engineering. Xylem consists primarily of tracheids, cells that die at maturity. After completing growth and development, the living contents disintegrate, leaving only the cell wall behind. This creates hollow tubes, and many such tubes joined end to end form continuous conduits extending from roots to leaves. Water moves through these dead tubes, pulled upward by the evaporation of water from leaves. The walls of tracheids must withstand the tension created by this pulling force without collapsing. Yet they must also allow water to move from one tracheid to the next through specialized structures called pits. True roots evolved in tandem with vascular tissue, providing both anchoring and resource acquisition. Unlike the simple rhizoids of bryophytes, which might be just single cells or simple filaments, true roots contain vascular tissue continuous with that in the stem. The tip of a growing root is protected by a root cap, covering the delicate growing point as the root pushes through soil. Behind the root cap, a zone of cell division called the apical meristem produces new cells that elongate and differentiate into the various tissue types of a mature root. The architecture of root systems varies greatly, reflecting different resource acquisition strategies. Some plants develop taproot systems, with one main root growing straight down and smaller lateral roots branching from it. Taproots can penetrate deep into soil, reaching water sources far below the surface. Desert plants often employ this strategy, with some species having roots extending 10 meters or more into the earth. Other plants develop fibrous root systems, with many thin roots spreading in upper soil layers. This architecture efficiently captures water from recent rain before it percolates deep into ground. Grasses typically have fibrous systems, their dense root networks preventing soil erosion. Specialized root modifications serve particular functions. Mangroves, growing where roots periodically submerge in salt water, have evolved aerial roots that project upward from muddy substrate into air. These pneumatophores allow oxygen acquisition in waterlogged soil. The association between plant roots and mycorrhizal fungi deserves special mention, as this partnership may have been crucial for initial land colonization. Approximately 80% of modern land plants form mycorrhizal associations. The fungi form networks of microscopic filaments extending from roots into surrounding soil, penetrating tiny spaces roots cannot reach, and extracting nutrients roots cannot efficiently absorb, particularly phosphorus. In exchange, plants provide fungi with sugars from photosynthesis. Lignin appears to have evolved somewhat later than the other major adaptations as plants began growing taller and faced increasing demands for structural support. This complex polymer, built from aromatic alcohols called monolignals, impregnates cell walls of vascular tissues and other supporting structures, providing rigidity that resists compression and bending. Unlike cellulose, which consists of sugar molecules linked in regular patterns, lignin forms through somewhat random coupling of its building blocks, creating a three-dimensional polymer network. This irregular structure makes lignin remarkably resistant to degradation. Lignin appears primarily in cell walls of xylem tissue and cells specialized for structural support. Its presence in xylem serves double duty, providing mechanical strength to prevent collapse under transpiration tension and contributing to overall stem rigidity. In trees, lignin makes up a substantial fraction of wood, contributing to its hardness and strength. The chemistry of lignin, once thought unique to land plants, has been discovered in some xerophytes and even in some seaweeds. The presence in seaweeds represents convergent evolution, different lineages independently evolving similar solutions to similar structural problems. While not all land plants contain lignin, most vascular plants do. Bryophytes generally lack significant lignin, contributing to their characteristically soft, pliable tissues. This absence, combined with lack of true vascular tissue, explains why bryophytes remain small. They simply lack the materials needed to build tall, rigid structures. The capacity for continuous growth through apical meristems represents another crucial adaptation. These small zones of undifferentiated cells at shoot tips or root tips continue proliferating throughout the plant's life. Unlike animal growth, which typically ceases at maturity, plant growth continues indefinitely through these meristems. At root tips, apical meristems produce new cells that allow roots to extend deeper into soil, searching for water and nutrients. At shoot tips, they enable stems to grow taller and branches to extend, elevating photosynthetic surfaces higher in competition for light. This continuous growth capacity, coupled with structural support from lignin and transport capabilities of vascular tissue, enabled plants to achieve sizes impossible in their ancestors. Protection of reproductive cells required the evolution of sporopollenin, a tough polymer characterized by long chains of organic molecules related to fatty acids and carotenoids. This substance, extraordinarily resistant to chemical and biological degradation, became a crucial component of spore walls. Spores often travel through air, exposed to intense UV radiation during dispersal. Sporopollenin provides protection, shielding reproductive cells from radiation damage. Interestingly, sporopollenin has been found in some xerophyte algae, suggesting this protective mechanism evolved before complete transition to land, another example of pre-adaptation in aquatic ancestors. These structural innovations did not appear simultaneously. The fossil record and comparisons among living plants suggest they appeared in sequence over millions of years. The cuticle likely evolved first, appearing even in some aquatic ancestors where it may have served other functions before being co-opted for reducing water loss. Stomata appear next, arising as solution to the gas exchange problem created by the cuticle. Vascular tissue and true roots evolved together in the transition from bryophyte-like ancestors to early vascular plants. Lignin appears later, as plants began growing taller and faced increasing demands for structural support. Understanding these adaptations as components of an integrated system rather than isolated features illuminates how plants function. The adaptations work together, each enabling or enhancing the others. Stomata only make sense in context of the cuticle. Without a waterproof barrier, controllable pores would provide no advantage. Vascular tissue only works effectively with lignin for support. Soft tubes would collapse under their own weight in tall plants. Roots need vascular tissue to transport what they absorb. Without transport capability, deep roots would provide little benefit. The modular nature of these adaptations allowed plants to fine-tune them for different environments. Desert plants might develop especially thick cuticles, widely spaced stomata, and deep taproots, while rainforest plants might have thinner cuticles, abundant stomata, and shallow spreading root systems. Each combination represents a different solution to fundamental terrestrial challenges, optimized for particular environmental conditions. The structural adaptations also vary in how completely different plant groups employ them. Tree ferns possess vascular tissue and true roots, but lack the extensive lignification of seed plants. Their trunks gain support partly from overlapping leaf bases rather than woody tissue. Horsetails have hollow stems strengthened by silica deposits rather than extensive lignin. These variations demonstrate that multiple structural solutions to terrestrial challenges exist, and different lineages have explored different combinations. When we observe modern plant diversity through the lens of these structural innovations, patterns emerge. The distribution of bryophytes in moist habitats reflects their limited toolkit, their lack of vascular tissue, and significant lignification. The global success of ferns in understory environments reflects their vascular tissue and true roots, but continued dependence on water for reproduction. The dominance of seed plants in most terrestrial ecosystems reflects their complete toolkit, fully developed and elaborated over evolutionary time. The transition from aquatic to terrestrial life, viewed through structural adaptations, reveals not a single revolutionary leap, but a gradual accumulation of innovations. Some innovations appeared in aquatic ancestors as pre-adaptations, serving other functions before being co-opted for terrestrial challenges. Others evolved specifically in response to terrestrial conditions, refined over millions of years. Together, these structural features transformed a group of aquatic algae into the diverse array of land plants that cover our planet today, from tiny mosses to towering redwoods. Each visible feature, each microscopic structure, represents a solution to fundamental challenges of terrestrial existence, assembled into an integrated system that has proven extraordinarily successful across nearly 500 million years of plant evolution on land. Chapter 4. Two Lives in One. The life cycle of an organism describes the sequence of stages through which it passes from conception to the production of its own offspring. For most animals, including humans, this life cycle follows a relatively straightforward pattern. We exist primarily as diploid organisms, meaning our cells contain two sets of chromosomes, one inherited from each parent. Only our gametes, the sperm and egg cells, are haploid, containing just one set of chromosomes. These haploid gametes fuse during fertilization to restore the diploid state, and the resulting zygote develops into a new diploid individual. This pattern, called a diplontic life cycle, dominates the animal kingdom and seems intuitively natural to us. Plants, however, follow a fundamentally different pattern, one that can seem alien at first encounter. Most land plants exhibit what biologists call alternation of generations, a life cycle in which an organism alternates between two distinct multicellular forms. One form is haploid, with cells containing a single set of chromosomes. The other form is diploid, with cells containing two sets of chromosomes. Each form is not merely a stage of development within a single individual, but rather a separate, free-living organism with its own structure and function. This haplodiplontic life cycle represents one of the defining characteristics of land plants and has profound implications for how these organisms live, reproduce, and evolve. The haploid and diploid forms, while genetically related as parents and offspring, can look and function remarkably differently. The haploid multicellular form is called the gametophyte, a name derived from its function of producing gametes. The diploid multicellular form is called the sporophyte, named for its production of spores. These two organisms alternate in a regular cycle. The gametophyte produces gametes through mitosis, ordinary cell division that maintains the chromosome number. Two gametes, typically a sperm and an egg, fuse in fertilization to produce a diploid zygote. This zygote develops through mitotic divisions into the multicellular sporophyte. The sporophyte, in turn, produces haploid spores through meiosis, the special form of cell division that halves the chromosome number. These spores germinate and develop through mitotic divisions into new gametophytes, completing the cycle. The alternation between these two phases occurs in all groups of land plants, but the relative prominence of each phase varies dramatically across different lineages. This variation reveals an evolutionary trend that has profoundly shaped the diversity of plant life. In the most ancient lineages of land plants, the gametophyte dominates. The sporophyte remains small, short-lived, and nutritionally dependent on the gametophyte. In more recently evolved lineages, this relationship reverses. The sporophyte becomes the dominant, conspicuous phase, while the gametophyte shrinks to microscopic dimensions and brief existence. We will trace this progression from mosses through ferns to seed plants and also explore how plants evolved from producing one type of spore to producing two distinct types, a critical step toward the evolution of seeds. Consider first the bryophytes, that group of non-vascular plants including mosses, liverworts, and hornworts. When you observe a patch of moss growing on a tree or rock, the green, leafy structures you see are gametophytes. They represent the haploid generation, and they are what we typically think of as the moss plant. These gametophytes carry out photosynthesis, manufacture their own food, and can live for months or years. They represent the independent, self-sustaining phase of the life cycle. The moss gametophyte begins life as a single haploid spore that germinates when it lands in a suitably moist environment. The spore's outer wall breaks open, and the cell inside begins to divide and grow. In mosses, the young gametophyte initially develops as a filamentous structure called a protonema, a branching network of green threads that looks remarkably like a filamentous green alga. This resemblance reflects the aquatic ancestry of land plants. After a period of growth, the protonema produces buds that develop into the familiar leafy shoots of the mature gametophyte, with structures that carry out photosynthesis, while simple rhizoids anchor the plant to its substrate. When conditions are favorable, typically during moist periods, the moss gametophyte produces reproductive structures called gametangia. The male gametangium, the antheridium, produces numerous sperm cells, each equipped with 2 flagella that enable swimming through water. The female gametangium, the archegonium, has a distinctive flask shape with a swollen base called the venter containing a single egg cell, and a long neck through which sperm must travel. When rain or dew creates a film of water, antheridia release their sperm, which swim through the water film to reach archegonia. One sperm fuses with the egg in fertilization, creating a diploid zygote. The zygote, still enclosed within the protective venter of the archegonium, begins to divide and develop into the sporophyte generation. The moss sporophyte consists of a stalk called a seta, topped by a capsule called the sporangium. This structure grows upward from the tip of the gametophyte, eventually rising above it, but remains physically attached throughout its life, drawing water and nutrients from the gametophyte tissue. The sporophyte is not green and photosynthetic, except sometimes slightly so in the capsule. It depends entirely on the gametophyte for sustenance. Its purpose is singular, to produce and disperse spores. Inside the capsule, specialized cells called sporocytes undergo meiosis, producing thousands of haploid spores. When mature, the capsule opens through an elaborate mechanism, often involving cells that respond to humidity changes. As the capsule dries, tension tears it open or lifts off its lid, releasing spores that are carried away by air currents. Those fortunate enough to land in moist, favorable environments will germinate, developing into new gametophytes and completing the cycle. This pattern, where the gametophyte is large and independent while the sporophyte is small and dependent, represents what we might call the ancestral condition for land plants. The gametophyte, being haploid, more closely resembles the ancestral condition in charophyte algae. The evolution of a multicellular diploid sporophyte represented an innovation, a new phase inserted into the life cycle. And initially, this new phase remained small and subordinate. As we move to examine the ferns and other seedless vascular plants, we observe a dramatic shift. When you look at a fern growing in a forest, the large leafy plant you observe is the sporophyte. This represents a complete reversal from the moss condition. In ferns, the sporophyte has become the dominant, independent photosynthetic organism, while the gametophyte has been reduced to a small, often inconspicuous structure. The fern sporophyte possesses sophisticated structural adaptations. It has true roots that anchor it in soil and absorb water and minerals. It has true stems with vascular tissue that transports materials throughout the plant. It has true leaves, called fronds in ferns, that carry out photosynthesis. The sporophyte may live for years, or, in some tree ferns, for decades, growing larger season by season. The fern sporophyte produces spores in sporangia that typically develop on the undersides of fronds. In many ferns, these sporangia are grouped into clusters called sori, visible as brown patches on the lower surface of fertile fronds. Each sporangium contains sporocytes that undergo meiosis to produce haploid spores. When mature, the sporangium opens through an elegant mechanism involving a row of specialized cells called the annulus, which flings the spores away with surprising force. The fern spore that lands in a favorable location germinates into a gametophyte called a prothallus, typically a small, heart-shaped green structure, rarely more than a centimeter across. It is only one cell layer thick and lies flat against the substrate. The prothallus is photosynthetic and nutritionally independent, but it remains small and delicate. It produces both antheridia and archegonia, usually on the same individual. Like mosses, ferns require water for the sperm to swim from antheridia to archegonia. After fertilization, the resulting zygote develops into a young sporophyte while still embedded in the archegonium. The young sporophyte is initially dependent on the gametophyte for nutrition, but this dependency is brief. The sporophyte quickly develops its first root and its first leaf, which begins photosynthesis. Within weeks or months, it becomes nutritionally independent. The prothallus, its reproductive function complete, soon withers and dies. The sporophyte grows and matures over subsequent months or years, eventually producing its own spores. This reversal in dominance between mosses and ferns represents a major evolutionary transition. The shift from a dominant gametophyte to a dominant sporophyte brought several advantages. The diploid sporophyte, with 2 copies of each gene, possesses greater genetic flexibility than the haploid gametophyte. Harmful mutations in one gene copy can be masked by a functional copy on the homologous chromosome, allowing the organism to survive genetic damage that would be lethal in a haploid individual. This buffering effect permits diploid organisms to accumulate and maintain more genetic variation, providing raw material for evolutionary adaptation. The larger, more complex sporophyte can also grow taller, compete more effectively for light, and produce vastly more spores than the small moss sporophyte. These advantages help explain why vascular plants, with their dominant sporophytes, have been so successful in colonizing diverse terrestrial environments. The trend toward sporophyte dominance continues and reaches its extreme in the seed plants, which include both gymnosperms like pines and angiosperms like flowering plants. In these plants, the sporophyte becomes not just dominant, but essentially the only phase we normally observe. The gametophytes are reduced to microscopic structures that live briefly and depend entirely on the sporophyte for nutrition. Consider a pine tree, a familiar representative of the gymnosperms. The towering tree, with its woody trunk, needle-like leaves, and extensive root system, is the sporophyte. This organism may live for centuries, growing to heights of 30 or 40 meters or more. The gametophytes of the pine, in contrast, are so small and short-lived that most people never knowingly encounter them. Pine trees produce 2 different types of spores, a condition called heterospory, in contrast to the homospory of mosses and most ferns, which produce only one type of spore. The male spores, called microspores, are produced in male cones, the small, soft cones that appear in clusters and live only briefly. Inside structures called microsporangia, cells undergo meiosis to produce haploid microspores. Each microspore develops into a male gametophyte while still inside the cone. This male gametophyte, consisting of only a few cells, is what we recognize as a pollen grain. The female spores, called megaspores, are produced inside the large woody cones. These cones contain structures called ovules, and inside each ovule is a megasporangium where meiosis produces megaspores. Typically, only one of the 4 megaspores produced survives. This surviving megaspore develops into a female gametophyte that remains inside the ovule, never released, never independent. The female gametophyte consists of thousands of cells and produces several archegonia, each with an egg cell. When a pollen grain lands on a female cone, it germinates, producing a pollen tube that grows through the tissue of the ovule toward an archegonium. Sperm cells travel through this pollen tube to reach and fertilize the egg. The entire process from pollination to fertilization may take more than a year in pines. After fertilization, the zygote develops into an embryonic sporophyte still inside the ovule. The ovule develops into a seed, a package containing the embryonic sporophyte, nutritive tissue derived from the female gametophyte, and a protective seed coat derived from sporophyte tissue. In this cycle, the gametophytes have become so reduced that they are hardly recognizable as independent organisms. The male gametophyte is just a pollen grain, a few cells. The female gametophyte, while larger, never leaves the sporophyte that produced it and serves primarily as nutritive tissue for the developing embryo. The dominant sporophyte controls the entire life cycle, protecting and nourishing the tiny gametophytes. Flowering plants carry this trend even further. The male gametophyte of a flowering plant is even more reduced than that of a pine, consisting of just 3 cells by the time it is mature. The female gametophyte typically consists of only 7 cells. These microscopic gametophytes complete their reproductive functions in a matter of hours or days, then are consumed or incorporated into developing seeds. The massive, complex sporophyte, which may be a tiny herb or an enormous tree, dominates completely. The distinction between homospory and heterospory deserves additional attention, as it represents an important evolutionary innovation. Homosporous plants produce one type of spore that develops into a gametophyte capable of producing both male and female gametes. This is the ancestral condition, seen in mosses and most ferns. Heterosporous plants produce 2 types of spores, small microspores that develop into male gametophytes and larger megaspores that develop into female gametophytes. Heterospory evolved independently in several plant lineages and is characteristic of all seed plants. Heterospory brings several advantages. It allows for specialization, with male and female gametophytes optimizing for their different functions. It also enables the female gametophyte to accumulate more nutritive reserves, since megaspores are larger and can carry more stored resources. This proved crucial for the evolution of seeds, where the female gametophyte provides nutrition for the developing embryo. The fossil record shows that heterospory appeared in the ancestors of seed plants before seeds themselves evolved, suggesting that heterospory was a prerequisite for the later evolution of seeds. This evolutionary progression from dominant gametophyte to dominant sporophyte reflects a fundamental reorganization of the plant life cycle. Several factors drove this transition. The diploid condition of the sporophyte provides advantages in terms of genetic variation and the masking of harmful mutations. The larger sporophyte can produce more spores than a small sporophyte, increasing reproductive output. The increasing independence of reproduction from external water, culminating in the pollen-based fertilization of seed plants, required reducing the gametophyte phase that depends on water for fertilization. Understanding alternation of generations transforms how we view plants. That majestic oak tree in the park is not analogous to an adult human. Rather, it is analogous to one phase of a human life cycle, as if the diploid phase of human development somehow grew to enormous size and lived for centuries, while the haploid phase remained microscopic and brief. The moss growing on a rock is not equivalent to a fern. The moss you see is haploid, while the fern you observe is diploid, representing fundamentally different phases of their respective life cycles. This life cycle pattern, seemingly complex and counterintuitive to those familiar only with animals, has proven remarkably successful for plants. It provides flexibility, allowing evolutionary changes in the relative prominence of each phase to suit different environmental challenges and opportunities. The alternation between gametophyte and sporophyte, between haploid and diploid, between sexual and spore-based reproduction has enabled plants to colonize virtually every terrestrial environment on Earth. From the wettest rainforests to the driest deserts, from Arctic tundra to tropical islands, this dual-phase life cycle continues to shape how plants live, grow, and reproduce, demonstrating that there is more than one way to organize a complex multicellular life. The colonization of land by plants stands as one of evolution's greatest achievements, fundamentally reshaping our planet over 470 million years. Through innovations like the protective cuticle, gas-regulating stomata, vascular systems, and lignified tissues, plants conquered the challenges of desiccation, radiation, gravity, and reproduction without water. Their unique life cycle, alternating between gametophyte and sporophyte generations, provided flexibility to adapt across diverse environments. This transition created the foundation for all terrestrial ecosystems. The oxygen we breathe, the soil beneath our feet, the food chain sustaining land-dwelling life, all trace back to those first plants that ventured from water onto barren rock. As you encounter any green plant, remember it embodies hundreds of millions of years of evolutionary innovation, testament to life's extraordinary capacity to transform profound challenges into opportunities that would reshape an entire world. Thank you for listening.